Thank you all for being here. It's my pleasure to moderate this workshop and to introduce our speakers today. So just to mention, I've started the recording and we'll be sharing the recording after the session, but we'll probably exclude the question and answer period for privacy reasons. So I think, Roger, in your absence, we decided to go with the order on the poster you're seeing. So we'll start with David, Alex, and then Roger. So hi, everyone. Thanks for attending this session. I know there's a lot of exciting things happening right now. So thank you for choosing to join us. This Block 3 workshop is titled Roles of Academia and Industry in Psychedelic Research, and my name is Aleksandrova, and I'm a postdoc in psychiatry at the University of British Columbia, and the coordinator for the MATRIX-N Research Cluster, which organized this event. And it's my pleasure to moderate this workshop and to welcome you all. And I would like to briefly acknowledge that UBC and Vancouver are situated on the traditional, ancestral, and unceded territories of the Coast Salish peoples, including the Musqueam, Squamish, and Tsleil-Waututh nations. And I'm privileged to reside, learn, and work on these beautiful lands. And I know that you're all joining us today from many places near and far, so I encourage you to take a moment to acknowledge the traditional owners and caretakers of these lands. And I also recognize that psychedelic-containing medicines have been used by Indigenous peoples in healing ceremonies and cultural practices since time immemorial, and that acknowledging Indigenous traditions and knowledge is necessary within the psychedelic research community. And this needs to go hand in hand with attempts to practice reciprocity with Indigenous people and to make research more inclusive. So now it's my pleasure to introduce our three speakers today, Dr. David Olson, Dr. Alex Kwan, and Dr. Roger McIntyre. All three have high impact academic research programs related to psychedelics and ketamine, and we'll discuss how their academic work has led to translational efforts at companies or to industry funded research. So to begin with, we'll have Dr. David Olson, who studied chemistry and neuroscience at Stanford University and the Stanley Center for Psychiatric Research at the Broad Institute of MIT and Harvard. He's a professor and currently serves as the director of the Institute for Psychedelics and Neurotherapeutics at the University of California, Davis. His expertise spans central nervous system medicinal chemistry, molecular and cellular neurobiology, and behavioral neuropharmacology, and his academic lab has made several important discoveries related to the mechanisms of psychedelics and related compounds. And in 2019, he co-founded Delix Therapeutics, a company developing non-hallucinogenic analogs of psychedelics for treating a variety of brain disorders. Next, we'll have Dr. Alex Kwan, who is an associate professor of biomedical engineering at Cornell University. His research focuses on the neurobiology of antidepressants, and he's particularly interested in using optical imaging methods to determine how drugs such as ketamine and psychedelics modify brain circuits. Alex is passionate about rigorous research and supports open science and sharing data. He actually grew up in Vancouver and received a Bachelor in Science at Simon Fraser University and then a PhD in Applied Physics at Cornell University. From 2013 to 2022, he was a faculty member in the Department of Psychiatry at the Yale School of Medicine. He's also a recipient of the 2022 One Mind Compass Pathways Rising Stars Awards, and has received an industry research grant. So I'm excited about learning more about that. And then finally, we have Dr. Roger McIntyre, who is currently a professor of psychiatry and pharmacology at the University of Toronto and the head of the mood disorder pharmacology unit at the University Health Network in Toronto. He's also the chairman and executive director of the Brain and Cognition Discovery Foundation, BCDF. Dr. McIntyre is one of the most recognized psychiatrists in the world in the area of mood disorders. He has extensive collaborations with private sector but not limited to, for the pharmaceutical industry, the insurance industry, and the healthcare industry in Canada, the United States, and globally. Dr. McIntyre is the founder of the Canadian Rapid Treatment Center of Excellence and the CEO of Braxia Scientific, focusing on ketamine and psychedelic clinical research. So I'm very excited to welcome all three speakers, and please drop any questions you have in the question and answer portion of the Whova platform on the right side of the screen. We'll have some time for audience questions after the presentations. So now I'd like to pass it on to Dr. David Olson, who should be able to share his slides.
Give me 1 second here. All right, how's that, Lily? Is it okay?
Go ahead.
Great. All right. It's good to be with all of you today. Before we get started, I want to mention that in addition to my academic appointments at UC Davis, I'm also a co-founder and the Chief Innovation Officer of Delix Therapeutics, which is relevant to the discussion today. So our academic group has been really interested in trying to find new treatments for a variety of different brain disorders, but we focused on conditions that involve changes in the structure of neurons, not only neurodegenerative diseases like Alzheimer's disease, but also the so-called stress-related neuropsychiatric diseases like depression, PTSD, or substance use disorder. And what I'm showing you up here is some cortical neurons that have atrophied in response to chronic stress, which is really a hallmark of many of these illnesses. Now, current treatments to address these conditions have really tried to rely on rectifying chemical imbalances in the brain through the use of things like selective serotonin reuptake inhibitors. And unfortunately, those types of medications have proven largely ineffective. SSRIs take weeks to months to demonstrate any kind of efficacy, and about a third of all patients will never respond to these treatments. And so our group has really been attempting to address this problem from a different angle. We've been trying to identify small molecules that can produce long-lasting changes in behavior by selectively modifying specific neural circuits. And we thought that one of the best ways to impact a neural circuit might be to physically change the structure of the neurons within one of those circuits. And so we spent a lot of time looking for molecules that we call psychoplastogens that can rapidly change the structure of neurons like these atrophied neurons in the prefrontal cortex. And so during our search for safe and efficacious psychoplastogens, we were led to the classic serotonergic psychedelics, and it feels like this was a lifetime ago. It's about half a decade ago now. We published a study showing that classic psychedelics like DMT and LSD are really good at promoting cortical neuron growth. And so you can compare this to the vehicle-treated neuron in the middle. You can see that the psychedelic-treated neurons have substantially more complex dendritic arbors. And if you do some super resolution microscopy and zoom in on one of these dendrites, you can see an increase in the number of dendritic spines and these filopodia-like structures. Now, something about psychedelics that is really unique is that they have not only rapid effects, but sustained effects after the drugs have been cleared from the body. So to demonstrate that these molecules can produce kind of lasting changes in neuronal structure, we relied on a very rapidly cleared psychedelic DMT. The half-life of DMT in a rodent is only about 10 minutes. And so for this experiment, we administered DMT via IP injection to a rat. We then waited 24 hours, a time period when all of the compound had been cleared from the body. We then sacrificed the animals, looked at neuronal structure via Golgi staining and function via electrophysiology. And we find that DMT produces this very large increase in dendritic spine density in the cortex. And this is accompanied by functional effects. We see an increase in both the amplitude and the frequency of these spontaneous excitatory postsynaptic currents. Now, I certainly believe that hallucinogenic psychoplastogens will be very useful for some patients. Our group recognized that the necessity of in-clinic administration, as well as a variety of other factors, limits the general scalability of these medicines. And so in order to broaden patient access, we started to develop these non-hallucinogenic psychoplastogens. And I'm just going to tell you about one of these molecules that we've discovered called tabernanthalog, or TBG. And so for this experiment, we're using these mice that have their neurons labeled with a fluorescent protein. And what that allows you to do is to put a window in the skull and use a 2-photon microscope to peer inside the brain. And you can look at a section of dendrite. And then what you can do is you can give an injection of TBG, wait 24 hours, come back and look at the exact same section of dendrite. And you can count how many spines were formed, like this one in blue, and how many spines were eliminated, like this one in red. And what we find is that TBG does not really impact spine elimination, but it does promote spine formation. And it does so to a comparable extent as the hallucinogenic drug DOI. Now, we believe that these changes in structural plasticity kind of manifest as broad therapeutic effects. And so we find that TBG has antidepressant-like effects in the forced swim test, comparable to ketamine. We also find that it has anti-addictive properties in a 2-bottle choice paradigm relevant to alcohol use disorder. We've also found that TBG seems to have some benefit for reducing heroin-seeking behavior. Now, as an academic, it's great when you make a discovery like this, but at the end of the day, what we really care about is helping patients. And so we really want to take this academic research and move it into the clinic so that people can actually benefit from these results. And so what I'm showing you here is a typical picture illustrating the so-called valley of death. On the left, you can see that there's a fair amount of funding for academic researchers to do what they do best, and that is to be highly innovative, to come up with new targets, new therapeutic strategies, new approaches to treating diseases. And once they've identified that, what has to happen is there has to be a handoff to industry, because industry is really good at reducing these things to practice, to actually create a product that can make it to the market and benefit patients. And where so many great ideas go to die is right here in this valley of death. And so what we've done at UC Davis to try to bridge the gap here between academic research and work done in industry is to create a new institute called the Institute for Psychedelics and Neurotherapeutics, or IPN. And we really see IPN as a catalyst for translating academic work into marketed products. And so the way the IPN works is we bring in funding to do basic psychedelic science through public grants, private philanthropy, and sponsored research agreements from industry. Then after we do some of this basic psychedelic science, we partner with all of the really incredible basic neuroscience groups at UC Davis, like the Mind Institute that focuses on neurodevelopmental conditions, or the Alzheimer's Disease Research Center, which focuses on neurodegenerative diseases. And we then take all of this knowledge and we use it to create novel intellectual property that we can then license to large pharma, small biotech companies, or spin out our own companies. And as an example of the last, I'm just going to show you Delix Therapeutics. This was a company that I co-founded. Delix has been growing very rapidly, and we're very proud of everything Delix has accomplished thus far. And we're hoping that Delix can take some of that UC Davis technology and actually get it into the medicine cabinets of patients. And with that, I'd like to thank these funding agencies on the right, these key collaborators in the middle, and most importantly, I'd like to thank all of the really, truly incredible, incredibly talented, hardworking, and creative scientists that I get to work with every day. Thank you.
Thanks, David, for this quick but a very informative introduction into your work. So again, direct any questions to the question and answer section on Kuva or to the Zoom chat, and we'll address as many as we can at the end. So next, I'd like to pass it to Dr. Alex Kwan. Alex, feel free to share your slides.
Okay. I should do it on the screen. Okay. Yeah.
Yeah.
So I'm glad to join this workshop to talk about the roles of academia and industry in psychedelic research. So my own primary affiliation is academic. I'm an associate professor in the Manning School of Biomedical Engineering at Cornell. I have some industry ties. You see a couple of disclosures. I'm a scientific advisor for 2 startup companies. in the psychedelic space, Empyrean Neuroscience and Freedom Biosciences. And I've also consulted for Biohaven Pharmaceuticals. So I'll talk a little bit about just broadly about what my lab is about and the research that we do. Then I'll actually go through a little bit of how we got academic funding to do most of this work. And then finally, I think as Lily alluded to, I'll talk about one example of an industry-sponsored research that our lab has recently completed. So broadly speaking, in my lab, we're very interested in psychoactive drugs. We're very curious how these small molecules can get in the brain, changes the structure and functions of neural circuits, and then lead to some fairly profound changes in behavior. And our goal is to be able to take some of these circuit level insight to be able to predict how new drugs might work, what might they then to do to neural circuits, and then to also predict what their behavioral effects might be. So this has obvious implications for things like drug discovery and drug evaluation. And the main method in my lab is microscopic optical imaging. So on the left is the two-photon microscope that David also talked about and also his lab uses. This is one that's actually in my lab right now, in the basement right now in Cornell. On the right is a mouse on such a microscope. So you can see the objective, the high numerical aperture objective on top of it. I think a key point to our research is the mouse is awake. So it's mimicking, I think, a good condition to test the drug action. And you can see this mouse is running on a ball. It's quite comfortable. Some of the mice would sit. And then this particular mouse is also actually licking and drinking water while, again, we're imaging the drug's effect microscopically. So if you come to our lab, this is the kind of image that you might see under one of these two-photon microscope. On the left is a cartoon of a primary neuron in a cortex. We're very interested in the dendrites out in the top, the apical trunk, which is where the neuron received a lot of the inputs. Here we're doing a Z-stack going into the cortex. And what you see is you can see the dendritic branches of this neuron out in the apical tree, right? And you can see all those tiny little knobs, which is also what David talked about, the dendritic spines, which is where this neuron receives the input, excitatory input from other neurons. So not only can we do structural imaging, but we can also look at dynamical signal using these kind of optical methods, again, microscopically. So here, instead of labeling the neuron with a static fluorophore, we can also label it with a calcium indicator. So here, whenever the dendrite lights up, it's because it has a calcium influx. So it actually tells us more about what's going on in terms of the biochemical signaling that might be changing due to drug action. So using these type of method, my lab has published now, I think, a series of studies looking at different kinds of drugs and their particular effects on dendrites using these imaging methods. I'll just highlight two examples. One example published last year, we look at the psychedelic psilocybin. We give mice single dose of either psilocybin or saline. And then we found that the dendritic spine density increased quite rapidly the day after when we administer it. And then it's also quite long lasting up to day 34. So this shows that psilocybin can induce fairly rapid synaptic rewiring in the brain, and it's at a very long time scale. If you look at the dendritic spine size changes, it's actually a different picture where you also see that transient increase of about 10%, but the time course is quite different. You can see it return to the baseline. So this kind of imaging really reveals some key feature for how this drug works, at least in terms of its action on neuronal morphology and circuit connectivity. As another example, here we're now leveraging that dynamic aspect of the imaging to look at calcium signaling. We're studying ketamine here. In this study, we look at the effect of ketamine before and after when the drug is still on board and what it does to calcium signaling in the dendritic spines. So you can see for saline, nothing much has changed, but for ketamine, it leads to a very robust increase in calcium transient in these dendritic spine compartments. You see more events, so there's an increase in frequency, but the events are also bigger, so you also see an increase in amplitude. So, again, I think these are sort of key feature or signature for how these drugs act on the brain, and ultimately, we think these are useful feature to look for if you're interested in developing drugs that might have similar properties, but perhaps without side effects. So actually, this participating in this workshop is quite instructive for me because I made this particular slide thinking back about how some of these research started and then also get funded. So my lab started 10 years ago at Yale, and then we recently moved to Cornell. On the top, I'm going to plot all the grants and research that happened with ketamine. On the bottom is the psychedelics. Ketamine work really benefit a lot from academic funding. a lot of it from NIH. But I do have to say it actually started from a smaller grant, which is a NASA Young Investigator grant from Brain and Behavior Research Foundation. Then we got the R21, which is a smaller version of the grant. And then 2 years ago, we got an R01 to study ketamine. If you look at psychedelic, it's actually quite similar, although it started later. Our interest in this area started about 4 years ago. We started with some pilot internal funding from Yale that eventually then lead to a R01 this year, as well as some foundation money now from One Mind. What's most interesting is I think about how overlaying this with publication. Here, red shows some primary research publication and purple shows some review articles. And you can see, I think, I definitely appreciate now those use of the smaller grant that really pushed the research forward. So NASA funded us before our first ketamine paper, and similarly, that pilot funding from Yale was really instrumental to allow us to even start that psilocybin study. So I think overall, and we are happy to discuss this more in the workshop later in the question and answer, but I think these small grants have been quite essential for starting the research, although eventually definitely getting federal support is possible. So switching gear, I will talk a bit about one recent industry sponsored research that we completed. So this is a work funded by Freedom Biosciences. I'm actually not the lead PI. The lead PI here is Chris Pittinger, who's a professor at Yale. But he mentored a psychiatry resident, Sarah Jefferson, who actually did most of that work in my lab. So here, Sarah is quite interested in the compound 5-MeO-DMT, 5-methoxy-DMT, which is a short-acting psychedelic. It's naturally found in the Sonoran Desert toad. It's interesting because this compound induces a psychedelic effect that's somewhat distinct from other compounds like psilocybin. The experience tend to be very intense. And as I mentioned already, it's quite short acting in the order of tens of minutes. So their potential may be more applicable for clinical use. So in this project, what we did was we started with looking at the head twitch response. This is work from an undergraduate student in the lab, Mark Dibbs. He put magnets in the mouse's ear, and then this allows us to put them in wire coils, which allow automatic detection of the head twitch response. So we can map these things over a long time scale and over a large cohort of mice. And I hope these curves, you can appreciate the dose dependence response of the 5-methoxy-DMT. You can see as we increase the dose from 5 to 10 to 20 to 40 meg per kg, you can see the increase in head twitch response. But no matter what dose we test for the 5-methoxy-DMT, you can see that the duration of that acute action is consistently shorter than that of the psilocybin, of the typical dose that we have used in the previous study. So that acute duration of action is not only seen in human, but also seems to be quite true in mice as well. And then for this particular project, the company was quite interested in the plasticity potential, again, leveraging some of these imaging tools to look at the turnover of dendritic spine as a potential biomarker for whether this drug might be promising. So Sarah did imaging a couple times before drug administration, and then several times more times after, up to 1 month later. And what she found is there's some similarity to psilocybin and also some differences. The similarity come from this effect on dendritic spine density, which is thought to be a structural substrate for some of the changes behavioral changes as seen in psilocybin. You can see that increase in the dendritic spine density relative to the vehicle, and it's also quite sustained. This was one of the questions that we had going in, whether the shorter acute effect might also mean shorter lasting plasticity, but here it seems like it could also last for quite a long time. Quite interestingly, when we look at the dendritic spine size, Joe, that doesn't change. That's reflective of the synaptic strength, and that seems to be quite flat between the two conditions. So again, it highlights some differences between these compounds. So that's all I have. But I just want to go back to the title slide to show, the involvement in industry can come in different flavor. We have done some sponsored research. I've also worked in a capacity as a scientific advisor or consultant. Yeah, happy to chat more in the discussion session.
Thanks, Alex. We're delighted to learn more about your recent research. And again, any questions for Alex or anyone else, please enter into the question and answer tab or into the chat. And finally, I'd like to welcome our final speaker, Dr. Roger McIntyre. Yeah, we see your slide.
Okay, can you hear me okay?
Yes, please go ahead.
Okay, hopefully I was at an airport. Hopefully it's not too noisy in the background. I'll keep my remarks pithy, just speaking to what I understood to be the question, that being the interface of academia and industry. I've been 25 years in academia, and I've also been an entrepreneur. I'm CEO of a company, a publicly traded company that endeavors to provide access to people who need mental health related treatment, but also to discover treatments. I'm a pharmacologist and a psychiatrist by training, and I've been drug discovery and development for 25 years. And really the message is that industry is where all psychiatric drugs were fully initially began and originated. And given the resources that are required to take products to market, no product could really get to market within the private sector. And I'll just go through some of this with you. For those less familiar, the antidepressant era began about 60 years ago, and it began with ushering in of the so-called monoamine hypothesis. The first antidepressants were developed by the private sector, a company called Ciba-Geigy that was rolled up by a company called Novartis. And the first antipsychotic was first developed in 1958. So the first antidepressant was 1957. The first antipsychotic was 1958, a company called Wander. And the first benzodiazepine or anxiolytic was developed by Hoffman LaRoche in 1955. So in the 50s, we had many different pharmaceutical companies, US and Europe based, that ushered in the development of these products. Moreover, the original discovery of psychedelics, whether it be LSD or psilocybin, was entirely sponsored by the pharmaceutical industry, that being Sandoz Pharma in Switzerland and other companies in Switzerland at the time. And more recently, ketamine, which has now appeared in our landscape, it was approved by the FDA in 1970, was developed by Upjohn Pharmaceuticals, which was merged with Pfizer Pharmaceuticals. So all of the drugs, that is the classic antidepressants, the more typical so-called psychedelics, that being psilocybin and ketamine, that are receiving the most attention to mood disorders, were all developed by pharma. Many might not be aware of the fact that ecstasy was first originated back in 1912 by Merck Pharmaceuticals. Merck developed ecstasy for the purpose of clotting blood. It didn't work very well to clot people's blood who had bleeding disorders. It kind of sat on the shelf and then got rediscovered later. So all of the drugs that we talk about in contemporary psychiatry and more investigational drugs, vis-a-vis psychedelics, all of them, not one exception, originated in the private sector. Now, what you have is you have academics who work in consultation, they're on payroll, they may work directly with the company, who provide advice, but the infrastructure, the scale, the ability to resource these developments could never really be done by academia on its own. Hence, in fact, we have the private sector. Now, it's a longer conversation about how we develop drugs. Developing drugs is not simply about having a concept and putting it in animals and showing that it works in humans. It's all about in fact, working very closely with regulators, and regulators very much determine how clinical trial design will take place. These conversations are also highly influenced by the reimbursement market. This is capitalism, and drugs are not usually developed unless there's a business case to develop them. That then, in fact, dovetails into the world of psychedelics, and one of the big questions we're struggling right now is with implementation science. In other words, how are these treatments going to be paid for, given the fact that we currently have in the United States, where I spend most of my time, about 5,000 less mental health care providers than we currently need, projected to be, 5 million, projected to be 10 million in the next 3 to 5 years. We clearly need to find ways to leverage the technology. How is this all going to happen at the scale that we need to actually offer it? Now, there's different ways we can develop treatments. People often focus in on the drug. Clearly, the drug is what matters, but actually it's the third issue. The first issue is, what is the patient population? That is the most important question. And can you suss out what is the therapeutic target and is it druggable? The second question is what are the measures? So one way to think about targeting treatments is, what are the dimensions of psychopathology? Now, historically in drug development, we would develop drugs for schizophrenia or depression or bipolar disorder or PTSD, and we still do. But now, in fact, there's an interest and there's regulatory buy-in from the European authorities, the American authorities, et cetera, to develop drugs that are targeting dimensions. And there's different biobehavioral matrices. One's called HiTOP. This is the RDoC, providing a framework for more precise characterization of the neurobiology, which is the first step towards so-called precision medicine. The next question, what are the measurements? You're only as good as the measurement. So when you're studying psychedelics, it's important to know, is it safe, does it work, and all that. But again, the first question is, what is the right population? What's the right outcome measure? And we haven't even fully determined that. This is a tool we developed, for example, in ketamine research, a tool that's highly sensitive to ketamine-based treatments. And these are some of the methodologic considerations that we need to really keep in mind. Now, as it relates to where we in fact need to go, these are not specific to any mental disorder. These are key unmet needs in psychiatry broadly, transdiagnostically. And there are many large, medium, and small pharmaceutical companies. Many are publicly traded, some are in the private sector, some are supported by private equity and venture capital enterprises and so on, that are endeavoring, in fact, to target one or more of these areas. And academia does not have the resources, nor does it have the human capital, frankly, in the business community, as well as the business development community, the R&D community, to be able to do all of this together. Just some rounding the bases. My colleagues have given you some very high science. I won't try and follow that incredible brain trust. They're obviously very, very versed on what they're providing for you, but there's many different directions that we're taking things. And given the vast complexity of neuroscience, it's important that we not just have the private sector, but we have in fact what we call co-opetition, where we have in fact various companies working together to try and in fact simultaneously target more than one of these systems, and we have many different ways we do that. You've heard a lot about ketamine. It's the first of the so-called rapid-acting, in quotes, psychedelics. It's a very imprecise moniker. This is just the first inning of the baseball game. There's many, many different derivatives of ketamine, which is 24 drugs in one. There's many other types of drugs that target ionotropic, metabotropic receptors. So again, this is all exciting. This is great in the classroom, but where the rubber meets the road is, can it be resourced? Is there a line of sight with respect to R&D? Is there a business case to be made? And can it be implemented? And these are aspects that are not typically taught in academic classrooms. The related question is, what's the route of delivery? Is it IV? Is it oral? Is it intranasal? Is it subcutaneous? These are all important questions to clinicians and patients and all of us, but it also has importance with respect to how we can develop these types of products. And there's different requirements with respect to how they'll be implemented, how they'll be reimbursed, what are the human resources, what's the personnel, what's the infrastructure. And all of these factors are as relevant as is the clinical science, the neuroscience, the pharmacologic science, but the implementation science. And it's the sheer vastness of this that requires the private sector and its resources to make this happen. It's also important we have guardrails, that we have guidance, we have clinical practice guidelines. And one of the concerns I have, quite frankly, with psychedelics currently is the relative absence of really best practices in clinical practice guidelines. We published the world's first guideline on ketamine, on how to do this properly, safely, akin to, or aligned with good science and so on. But this is another area, in fact, that is important. And I would say that academia probably plays more of a role on this side, the so-called knowledge translation. So when it comes to knowledge really generation, in theory, the classroom, but where it really takes off is in the private sector. But on the knowledge implementation and the knowledge translation, I think that's really where academia really steps up. Psilocybin looks promising. We'll see where it goes. This was the study last year. Compass read out their study. They're a public company that's quite well funded right now, going through their phase 3. We'll look forward to see how things pan out with that. But at the end of the day, we're still going to have big questions on implementation and reimbursement. They're going to have to be critical. So I started the company Braxia Scientific with an aim to develop treatments that are innovative, but also treatments that people can access. So Braxia is not only developing treatments, but is integrating technology, telehealth, telemedicine in its development protocols as a way of bridging gaps, this huge gap between the need and what's available. At the same time, we're also, in fact, involved in very early asset development. And again, the aim is, in fact, to find assets that are in phase 2, and we're a late stage company that takes treatments and then develops them, takes them across the so-called finish line in an accelerated fast track status. So I'll just keep it short. It's a workshop. There's my Twitter account. I post articles related to the topic for those interested. I'll keep this up for a moment. I think the workshop's open for discussion.
Thanks, Roger. Thank you all for your brief presentations that set up the stage for the discussion period. So before we dive into audience questions, maybe I could start with one, and it's pretty at the core of what this workshop is about. So based on your experience, how are successful academic and industry partnerships built? And how can universities encourage innovation and translation of research findings into health solutions through collaboration with the private sector? So we can maybe start with Roger first.
Yeah, so what this comes down to, it comes down to a recognition of what the objective is of the pharmaceutical industry. The primary objective of the pharmaceutical industry is to generate a profit. Now, we're in a very interesting time. We go in and out of philosophical sort of tides. Are we in a time of shareholder capitalism, or is it stakeholder capitalism? We've moved towards this more so-called stakeholder capitalism, where some would take the position that the purpose of the private sector is in fact to provide governance and social responsibility and things of this nature. That's a very active debate. At the end of the day, others would say that the purpose of business is business. That's the famous Chicago School statement. And at the end of the day, unless a company is generating profit, they will no longer be a company. So companies, despite the fact that there's been a shift towards more so-called stakeholder capitalism away from shareholder capitalism, the reality is you have to make money to pay the bills. So therefore, it is a profit industry. And it's clear that industry is open to partnerships, but like most partnerships, there has to be a kind of reciprocity of benefit. In other words, what is in it for them? What's in it for academia? And there's many examples, many. S-ketamine's a great example. S-ketamine was approved by the FDA in 2019. S-ketamine's been around for a long, long time. And a private company that has a joint venture with Yale University took this product, which was in part developed at NIH, and through this public-private partnership through Yale and its commercialization division, one of its commercialization units, then took S-ketamine and matured it, developed it to late phase 2, and then Johnson & Johnson then came in, saw the business opportunity, and then took it across the finish line. So I think, in fact, frankly, it begins with what is the role? The role of industry is to stay alive and to be self-preserving. You need a profit to do that. Secondly, I think, in fact, where at least in central nervous system in psychiatry, big pharma has really vacated early R&D. Big pharma is more into late stage, mid-stage, late-stage development. And so they're shopping around. This is a great time for colleagues like my colleagues on this workshop with me, sharp, smart people, to develop assets and to develop them to maybe phase 2, early phase 2, and companies will be shopping around. There's opportunities for academics, I think, in fact, to really show and tell their wares, if you will. And this is a unique time because if there is, in fact, an attractive option, if there's a business opportunity that's created by the company, there's all kinds of I think, lines of sight for people. So I'll just maybe stop there.
Thanks, Roger. Alex?
I don't have much to add. Actually, I'm just going to pass it to David because I think he's a very good example in making it working,
Sure, David.
So I think that in order to have a successful academic industrial partnership, I think the two parties really need to consider what the other party is looking for and look at it from their point of view. I think that's really critical. And what you find is a lot of times there'll be a lot of like heads butting because the goal of academic scientists are to publish. And that is not necessarily the goal of an industrial scientist, but there are places that you can meet in the middle. And in my opinion, some of the most successful academic industry partnerships has really been where industry has been funding basic research to move the field forward, which allows the academics to still be able to publish. And of course, the companies can use that information for a variety of their programs. But where I think sometimes it gets a little challenging is when companies want to keep things secretive. They don't want the information to get into the public sphere. As a public university, the University of California has to publish whatever we do. And so that's something that we really consider. The other side of this I think you asked about, how can universities encourage this type of collaboration? And I think one of the ways is to not demonize it. There is the tendency in academia to kind of look down on academics who are involved in industry partnerships. And I'll give you a perfect example without using any names. One of my colleagues is a serial entrepreneur. He's been incredibly successful. He has multiple companies that are actually impacting patients' lives. Almost all of his academic funding comes from industry. Now, he can support a lab of 50 people with that industrial support. But when he goes up for tenure and promotion, colleagues will say things like, he doesn't have enough NIH grants. All of his money comes from industry. And so I think that's missing the point, that he's pulling in money to do basic research, and that basic research is going to companies that are making impacts. And I think that we need to value the translational impact of academic science in the promotion process.
Yeah, that makes a lot of sense. Thanks for sharing your perspectives. I guess one last question from me before I dive into the audience ones. With so many psychedelic companies, all with different business models and, forming, and some of them already going out of business, what are some characteristics of a good psychedelic company, business, science, and social impact wise? Or in other words, what values do you try to incorporate into your businesses or look for in industry collaborators? Maybe Alex first.
Yeah, so for collaboration, since we started working in this area, we have been approached by different kinds of company, right, who wants to work together. And it's something that definitely, echoing what David has said, it has to also make sense for us in terms of the scientific question that we also want to address versus, maybe some specific compound that they want to test. And I think some of the key aspect of the company that I look for is they should have a very strong leadership. I think the people at the end is the key. They have to have, the right, I think the kind of pharmaceutical veterans in there, have to have the right scientists there. I think the existing research have to be solid as well. So I think ultimately, even if it's just they can provide a lot of funding, a lot of money, ultimately we also still want to waste the time to do this work if it is not going to actually lead to some good science. So I think those are a couple of characteristics I look for.
David?
What is a good company? Well, I think, like Roger said, from a business perspective, you've got to have a company that ultimately will be profitable. That's a given. As far as the science goes, I always think that, the role of especially new companies is to innovate, to do something substantially different than what has been done before. And so that's something that I always look for. Now, in terms of social wise, as far as like building a company, I think it's really important to think very hard about culture. And I would say that, of all of the accomplishments that Delix has had, the thing that I'm most proud of is that they won the ACE Award from the Healthcare Businesswomen's Association, because Delix has really put a lot of effort about thinking about creating a culture that really values diversity, equity, and inclusion, and really promotes the advancement of all people. And a large portion of those people are women. And so we actually have gender parity within our company, even within our C-suite, which is very unusual for most companies. But I think that's something that companies should be thinking more about now.
Thanks, Roger.
Many active ingredients. You need good leadership. You need to have a good business model, and you need to be resourced. So you need human capital, you need a business model, and you need liquid capital in the bank. And that's what is the pillars of getting a company going and going forward. And there's been very few companies that have been successful who didn't have most of those three. And of the various companies I think you're making reference to, many of these are publicly traded, many are privately traded. Most won't be around in 3 to 5 years. Probably 75% plus will be gone. In currency, and there was about a year and a half ago, 2 years ago, there was a real tide of funding and resource coming to this area. Global equities have really taken a downdraft over the last year. This particular space has become much more difficult to raise money in. And so you're going to see tremendous consolidation. And by the way, that's what happens in every space. Back in about 1920, if you can believe it, we had over 100 car companies in America. 15 years later, we had 3. So it's very common in development, in industry, to have a Gartner hype cycle where you have tremendous wealth come in, the tide comes in, there's tremendous hype. And again, the human capital, the business model, and of course having the leadership to direct it through doesn't always guarantee success, but you will not have success without those 3. And really, in fact, what happens is that you'll have a few of these companies will emerge as being the ones that are succeeding, and then that will then fuel forward a virtuous cycle for those companies.
Thank you all for sharing your insights. Now I'm going to take up an audience question. This is from Aleksandrova Jager. She says, thanks everyone, great talks. In the world of therapeutic intervention for mental health, where lots of psychedelics are being placed moving forward with psychedelic assisted therapy, I hear a lot of clinicians saying things like, I don't care how it works, only that it works. Why is it important to understand how these molecules work in the brain? Is it important for better care for patients? Does this thinking impair the efforts of bridging the valley of death?
Well, I think, look, I mean, I'll take the first stab at that. I think that our colleague who submitted that question, I think, makes a good point. I think at the end of the day, when you drive your car off the car lot, is it important that you have a mechanics level of expertise how a car works? Probably not. As long as the engine turns on, it goes into drive, and the left turn, right turn blinkers work and all that. So to some extent, I think the point's well taken. At the same time, at the clinical level, it is important to have some knowledge of these issues because there could be potential for interactions with other medications. So at the very least, you have to have some basic pharmacologic understanding of these medicines. I think it's a fair point as well that really sort of unraveling the precise pharmacodynamics and underlying mechanisms is probably more of interest to the academic, to the scientist, to the drug discovery, to the medicinal chemist, than it is to the doctor, the nurse, the physician assistant who's seen 65 patients today. So point well taken, but I still think it's clinically relevant insofar as safety and rational pharmacology and prescribing. But I think the point's well taken.
Just to follow up on Roger's analogy, one of the reasons I think it's important to understand how these drugs work is so that we can innovate and move past them. You don't need to know how a horse and buggy works in order to use it, but if you don't understand how it works, you can never evolve to produce an automobile. You're going to be doomed to be in the horse and buggy era forever. And so if we want to think beyond psychedelics, something 20, 30 years from now, what do the new treatments look like? You really have to understand how these things work in the brain.
Alex, do you have anything to add?
Yeah, I was about to say the same thing, that I think knowing how they work, in my mind, is a step towards further drug discovery. I think one of the issues with psychiatric drug has been the lack of that understanding, which leads to the lack of new drugs in this field. Thankfully now we just begin to have another 2 or 3 recently approved treatment, and I think it is a very important time to try to understand some of these mechanisms of action so we can actually leverage that to develop further new medications.
We have a question here from Dr. Anthony Phillips. And this is, I guess, related to something that was mentioned, but I hope it's still relevant. So are public-private partnerships, as just described for esketamine, a realistic option for sharing the benefits and profits of academic-based discovery?
The short answer is yes. Though to be very clear, esketamine was not developed by academics. It was actually incubated by an academic private-public partnership. Esketamine was developed by Pharmacia. But I think what I'm getting at is, and I think the short answer is yes, Big Pharma has left early R&D. It's too expensive. There's too many failures. It's not a good business to be in. So pharma's changed the business model where they are attempting to acquire products that have been developed by agencies broadly defined in the public or private sector. So historically, the way drugs were developed in psychiatry, all major drugs in psychiatry were all developed by big pharma, not academia. And the way it worked is that pharma had hundreds of PhDs and scientists and medicinal chemists busy in the lab finding the next greatest blockbuster. And then they went right from phase animal all the way through phase 1, 2, 3 to FDA approval. That's not the way it works anymore because companies have vacated early R&D. And that's where academia could come in. Insofar as developing assets that have some minimal viable product and so on. The challenge, though, is that there are exceptions, but the challenge is that most departments of neuroscience, departments of psychiatry, don't have the business aptitude, don't have the business development expertise. And I think, in fact, it's would be hubris to say, well, look, I'm an expert on a serotonin receptor, therefore I'm an expert in business development, I'm an expert in regulatory lines of sight and so on. And unfortunately, most departments, academic departments, are not equipped with the expertise to do that. Notwithstanding, yes, there are opportunities. And I think the greatest public-private partnership of all time occurred 2 years ago with the development of the mRNA vaccines, probably the best of all time. So that type of Operation Warp Speed could be adopted in psychiatry, and there's an appetite for these types of things, absolutely.
One other thing I want to mention to follow on with what Roger said is that academic scientists are not trained to think like industrial scientists. And that's something I think we really need to do a better job of. Something as simple as thinking about intellectual property. A lot of academics don't realize that they need to protect their inventions before they publish. Otherwise, no company is going to bring that forward. And that is a very simple thing that we can do early on, even in graduate training, to teach scientists how to protect these things so that, the public can ultimately benefit from them. Now, one other thing I think I'll say is, it's rare for an academic discovery to go all the way to the clinic, but when it does, it can be very profitable for the academic institutions. And I'll give you one example. Lyrica was developed by a medicinal chemist at Northwestern, and the royalties from Pfizer from Lyrica now make up something like 20% of the university's endowment. They've used that money to do a lot of good for the university too. They've reinvested that into, scholarships for undergraduate students, new buildings to do science. They brought in really great scientists to improve their departments. And so these types of collaborations can be hugely beneficial for academia.
I said that, should I move on, or do you have anything to add, Alex?
No, I'm good. I think they cover it well.
Mm-hmm. Okay, one question that we actually posed to the audience was, one second, can industry involvement and corporate interests introduce bias or other issues in the research process? And while you share your insights, I'll share the audience results. Maybe Alex?
Yeah, I think that is definitely one danger that I think the researcher have to think about. I mean, I think the company, when they engage in a partnership or sponsored research with academics, they typically have certain results in mind. I mean, they have their compound they want to test. They want to see a positive result. Yeah, so I think that's definitely a danger. I think in academics side of things, you're somewhat, I think, protected from the university side, where the university stipulate that any result has to be able to publish, and depending on, and they obviously have their own lawyers that can go through the contract and make sure that you're not bound to the whims of the company. But there are also more subtle, I think, influences that I think the researcher have to be aware of. That's why I actually think it's important to start to have more federal funding and other kinds of academic resources to start studying these in terms of foundation money as well. I think the NIH is turning, and there are other private foundations like the One Mind that's starting to fund this area. I think those kind of grants that are not attached to any particular idea or any particular theory would be quite important if we want to do some very good basic science research.
Mm-hmm. Yeah, so the breakdown, and apologies, these were my answers, but like options, but it seems like there's bias in any other field, or at least that's what most people seem to think. David or Roger, do you want to add anything to this question?
Yeah, I mean, look, I think the question is axiomatic in the answer. The question, I think, is not is there bias. The question is how can biases be mitigated, managed? There's always biases. One point I want to emphasize is that there's no question that if a company sponsors a study that has a tailwind, if you will, a positive moderational effect on the outcome of the study, and that's a well-replicated finding. I would also, though, say in the same sentence that when industry conducts a study, the data have to be submitted to the regulator for full scrutiny. It's the law of the land. It's a federal offense not to do so. Academics don't. So I've conducted hundreds, literally, of studies in my career as an academic. Not one of my studies has ever been subject to any audit of any sort. I'm not looking to be audited, but academics are not audited. And peer review is not an auditing of your data, your quality, your data integrity, your best practices, your personnel competencies, and so on. But it is for industry. So said differently, that the paradox is despite the very bad public relations industry has, and in some ways you could say it's justified in some cases, and I would say I agree, but notwithstanding that very bad reputation they have with respect to aspects of ethics and integrity, in fact, they're held to a much higher standard with respect to accountability and data integrity, and academics are held to no standard. There's really no ongoing auditing of most academic research with respect to the integrity of the data. So in point of fact, when I look at academic research, I'm an academic, I look at industry research, I know full hand from my own personal experience that the industry research has been held to a much higher standard of accountability.
Very interesting. And David, do you have anything?
I agree with what Alex and Roger both have said. Additionally, I'd say that I just think, the potential for bias is always there, but that's why it's really important just to be transparent. So people need to disclose their affiliations and all that kind of thing. And also, I think just good practice for a lot of academics is to have the work replicated and extended by other people that are not involved in the company. So that's something that my group does all the time, is we'll send compounds out to many other people, let them, make their own judgments on whether or not it works. They might have no affiliation with the sponsoring organization. And I think that can be a way to improve rigor.
Mm-hmm. And just one final question, any practical tips on navigating relationships with industry? So what to look for in contract negotiation, managing conflicts of interest, managing intellectual property? Any final thoughts?
Well, this is where I get the most frustrated, because usually it's the university and the company arguing about contracts. And I think that it's important to make sure that academics can publish. But of course, like industrial partners sometimes want to keep some data secret. And so one solution, I can't do this at the University of California because we're a public university, but other private universities can do this. And that is to run a study with some controls that are well known in the literature, psilocybin, ketamine, et cetera. And if a company has a molecule that they don't want to publish on, you put that in the contract, that will not be published, but the control data set will, for instance. That's one opportunity. As far as the conflict of interest and conflict of commitment, these, usually there's some kind of regulatory body within a university that looks at this. If you have some kind of financial stake in a company, a lot of times what you can do is make one of your colleagues the PI of the grant. And so they're responsible for reporting to the company, not you, things like that.
Thank you. Alex or Roger?
Yeah, I think my own experience so far, talking and working some with industry is, I realize there's no free lunch. They want something from me, and I also want something from them. And I think at some point Roger also said that's how it is, and that has to be negotiated. And I think the expectation has to be set quite clearly in the very beginning, and then that just helped develop a very steady relationship from then on.
Mm-hmm. And Roger, any final thoughts before we end? He might already be on his flight. So thank you all for joining us, and apologies to Kino and to Shali. I didn't get to your questions. They were directed specifically to David and about non-hallucinogenic psychoplastogens, but I'm sure that you can read a paper and get some of these answers, or just follow up with me and I'll try to get them for you. And other than that, thank you all for attending. Thank you, David, Alex, and Roger, for lending your expertise and your insights from your longstanding careers in this field. And yeah, we'll just end the session now, and in 10 minutes, we'll just have the closing session for the conference. So thank you all so much. Bye.